My name is Ritwik Kumar and I'm part of the Weizen Lab. Today, I will discuss my work that focuses on characterizing prophages and clinical strains of E. coli. Bacteriophages, or phages, are viruses that exclusively infect bacteria. Lysogenic phages are a type of phage that integrate their genome into the host's genome in the form of a prophage upon infection. When the host experiences environmental stress, the prophages excise, replicate, and lyse their host, releasing their progenies into the environment. Phages are central in the fight against antibiotic-resistant bacteria. They provide an alternative treatment called phage therapy to infections that are impervious to conventional medications. Without intervention, these infections are on track to kill 10 million people annually by 2050, posing a significant threat to global health. Prophages have the potential to optimize phage therapy and antibiotic treatments, yet there's little understanding of the presence of phages and their characteristics, creating a knowledge gap in the treatment. This project ultimately contributes to the field of knowledge of prophages in a unique clinical case. I'm specifically interested in the prophage activity of clinical strains of antibiotic-resistant E. coli that have been isolated from urine and blood samples of infected babies. I utilized two approaches to determine the presence of prophages in my bacteria. I used the bioinformatic approach to predict their presence by analyzing sequenced bacterial genomes, and I confirmed their presence with prophage induction experiments using genotoxic stressors. We screened 60 clinical strains of E. coli and could predict 38 high-quality putative prophages within 25 strains. From the predicted phages, 2 strains showed induction signals after being exposed to genotoxic stress. My results indicated that there were likely inducible prophages in the strains E. coli O71U and E. coli O39. E. coli O71U exhibited really interesting bioinformatic characteristics, primarily that it fell under the Peduovirus genus, which are P2-like prophages. These prophages have an important clinical significance, as they've also been isolated from many opportunistic strains of E. coli and can provide insight into their characteristics and relevance. For example, E. coli O157, which is responsible for many foodborne disease outbreaks, have Peduovirus prophages that trigger a niche expansion within animal intestinal microbiomes. This discovery of a Peduovirus prophage in E. coli O71U expands the understanding of the effect of prophages on opportunistic bacteria and continues to propel us into an innovative age of medicine. Thank you.